Hello. So in this video, I'm back with the discussion on HIV/AIDS. So, what is HIV/AIDS? How it spreads? It is caused by what? All these aspects we'll be discussing here. All of you know that AIDS disease, which is actually acquired immunodeficiency syndrome. This disease is caused by a virus which is called human immunodeficiency virus (HIV). It is a retrovirus. What is a retrovirus? I'll be telling you. Before that, historical aspect is given in your book. So it was first reported in 1981, and till now 25 million people have died because of this disease worldwide. So you can imagine how dangerous this disease is, and there is no overall cure, but treatment can lead to prolongation of the life of a person. How this virus spreads from one person to another, that we have already discussed, or you have got it in class 9, like through sharing of infected syringes during drug abuse, or during blood transfusion, or during pregnancy from mother to the fetus through placenta, or through breast milk, or through unprotected sexual contact. All these are the different ways by which this virus, means there has to be contact of body fluids. So through contact of body fluids, this virus spreads from one person to another. Now, why it is called a retrovirus? It is called a retrovirus because the genetic material, virus, it has a protein coat and a genetic material. That is the simple structure of any virus. So the genetic material in case of HIV is an RNA. RNA is a primitive molecule compared to DNA. So those viruses which have RNA as the genetic material, they are called retroviruses. Let us have a look at the structure. Now, how HIV multiplies and all is given in this diagram, but this diagram is flawed. The reason is that actually HIV does not look like this. So I'll just show you how it looks like, its internal structure and how it multiplies. So you see this. So you have been seeing this image quite often nowadays in your television set, because the structure of coronavirus is like this. So all this coronavirus, SARS virus, HIV, all these are belonging to the same group of retroviruses, and they have such similar structure. So what is the internal structure of your HIV? Let's have a look. So as I told you, any virus, it has a protein coat. So those spikes that you can see, those are made up of glycoprotein, and the tip of these spikes, they are rounded. They are made up of docking glycoprotein. Now, what is docking? Docking is a process which means to join, very simply to go and join. For example, like when spaceships, they go to the space, they dock with the ISS, the International Space Station. So that part which will help in that joining, that is called docking, because this, with the help of this part, they will join with the host cell. And internally, and this is a phospholipid envelope, phospholipid, the membrane that you got, biphospholipid layer. So like that. And internally, when we talk about the genome or the genetic material, they have this spiral RNA molecule, and one very important and critical enzyme which they have, and that enzyme, they have two viral genome here, and that enzyme is reverse transcriptase enzyme. So reverse transcriptase helps in reverse transcription. What is transcription? You have learned in your other chapter. Transcription is a process from where, like, suppose there is the DNA molecule, and from the DNA molecule, when an RNA molecule will be synthesized, so synthesis of RNA molecule from DNA molecule is called transcription. Here the reverse takes place. From RNA, DNA will take place, and that is done by this particular enzyme, reverse transcriptase. So let us now see how HIV multiplies upon entering your system, and how it actually eventually destroys your immune system, upon which the person will meet with a very slow and painful death. So there is the cycle, if I can show you in an enlarged way. Okay. So first what happens, the HIV will come and it will dock with the host cell. This is the host cell. Now you can see one thing written here, CD4. CD4 is actually, you can say, these are all like macrophages, then your T cells, the lymphocytes, all the protective cells of your body, they are called the CD4. Okay, they have these membrane proteins. These membrane proteins acts as recognition site for HIV. So this HIV will come, and with the help of this recognition site, it will dock with the help of the docking proteins. So first it affects the macrophages. What are the macrophages? Macrophages are some amoeboid big size cells present in the areolar tissue. I discussed in the earlier video on immunity. So upon docking, then what happens? Slowly the membrane of the host cell will join with the envelope of the HIV. The entire virus will not enter the cell, that you should know. The virus only injects the genetic material. So all these things, the HIV RNA, reverse transcriptase, all these will be injected into the host cell. Upon getting injected into the host cell, this viral RNA, with the help of the reverse transcriptase enzyme, it will form viral DNA. So from the viral RNA, viral DNA will be formed, because the virus DNA has to come and attach with the host DNA, because for the virus to multiply, the virus has to take over the working mechanism of the host cell. And how it can overtake the working mechanism of the host cell? By hijacking the information center. And information center of any cell, you know, is the DNA. So in order to hijack this DNA, the viral DNA has to come and attach, but this virus is not having DNA. So what it will do? So from the RNA portion, with the help of this particular enzyme, it will form the viral DNA. This viral DNA will enter the nucleus of the CD4 cell. CD4, it can be your either the helper T cells or the macrophages, and it will get attached with the host DNA. Now the moment it gets attached with the host DNA, it has hijacked the mechanism of the host DNA. Now the host DNA, what it will do? It will start producing those proteins which will be required for formation of new viral particles. So they will produce, so from DNA, you know what is produced? RNA is produced, and from RNA respective proteins are formed. So this particular viral DNA, it will now produce viral RNA. Okay, this is transcription now. Now this RNA will slowly come out of the nucleus, and after coming out from the nucleus, it will reach to the side of the membrane of the host cell, and from this RNA some new proteins will be formed which will be required to synthesize the protein coat of the virus. So this RNA will get covered up, it will slowly, the host cell membrane will swell up, and with the help of the newly synthesized protein, it will take the form of the viral protein cover, having the viral RNA inside it, and slowly it will pinch out, just like lysosomes pinch out or vesicles pinch out from Golgi body. So it will pinch out, and new viral particle will be released. So this is how the virus will keep on multiplying, and when they multiply at a rapid rate, so this is basically what happens inside macrophages. Inside helper T cells also they do a similar way, and thereby destroying the helper T cells. Now how it damages your immune system? Now you see, macrophages is part of the immune system, you know that. They will engulf the cell and they will destroy, they will engulf the foreign body or the germs, and they will destroy the germ. So when your macrophages are damaged, so the germs will not be, your body will not be able to destroy the germs. Similarly, we know that the main protection in our body is provided, slightly dark here, okay, is provided by plasma cells. how plasma B cells, how they provide protection? They produce what is called antibodies. So, and these plasma B cells, they are activated by helper T cells. Now what happens is that when your these helper T cells will be affected by HIV, so these helper T cells will get damaged. So when helper T cells will get damaged, the plasma B cells cannot mature, and when plasma B cells cannot mature, antibodies will not be formed, and so that line of defense also gets destroyed. So in multiple ways, macrophages are destroyed, helper T cells are destroyed. So in multiple ways, what happens? Your immune system starts failing. So when your immune system starts failing, then you start suffering from different related diseases. So let's go back to what is there in your text. So I hope you have understood how the HIV multiplies and how it damages your immune system. What else is given in the text? Let us have a look. So that is what has been discussed here. After that, how can we actually test the presence of HIV? There are different testing procedure. One is ELISA, enzyme-linked immunosorbent assay. They always test for these tests are based on presence of these membrane proteins. So if you look into any YouTube video of ELISA, you will very easily understand, and that's very interesting also. Understanding the membrane proteins and signaling pathway is very critical, and that is the main research area where biological sciences are now working on. When we'll discuss cancer, I'll show you different signaling pathway by which cancer can happen in a cell whatsoever. So much detail is not there in your book. They have simply given the name of the test, and with the help of that test, HIV can be detected. You can look into the test. Is very simple to understand. I'll discuss in one video how ELISA works. How can you prevent AIDS? If you know how AIDS, HIV spreads, so you will be very well aware how to prevent it. So you have to avoid unprotected sexual contact, use of condoms in reproduction chapter you have got. Then a mother should be HIV free. That much of different at different levels people should be made aware. Then there should not be drug abuse. Sharing of needles should not be there. People should be educated. December 1 has been dedicated as National AIDS Day world over, and there is an agency called NACO, National AIDS Control Organization, which looks after all this, creating awareness among masses. And so all these are the different ways by which we can actually prevent HIV/AIDS, and different medicine. So many people ask, is there any treatment? There is no treatment as such, but the life expectancy of a patient can be increased by using some drugs which are called antiretroviral drugs, and that is called ART, and antiretroviral therapy, which is given in the community health centers. So this is all about HIV/AIDS. I hope you have understood. In the next video, I'll discuss about cancer. Thank you.